So I guess I need to define my title, climate smart, because my Brazilian colleague here doesn't know what that means. So all that means in this case is that we're using gene editing to match the genetics of the animal to fit the environment. So that's what I'm going to be talking about. I'm going to focus mostly on slick, which I'll describe that trait later, but there's other traits that could be involved in tropical adaptation as well. I won't get into those today. Okay, so just to set the scene, what are we trying to address? These are a slide that I've taken from Alfred DeVries, who's my program officer at the Gates Foundation. But it's basically to show that having an animal meet its realized potential in productivity versus its true potential is a combination of factors that are involved in increasing that. So that includes having proper veterinary care, forages, but also having those genetics matched within the environment in which it's raised. So we've tried to improve production by shortcutting a lot of time in tropical zones by importing animals that were developed in temperate zones. And they don't do very well. And what you get is this effect with shown in the graph here on your right, where you have a higher increase in greenhouse gas emissions relative to the protein produced. So some would say that these inefficiencies, 70% of them in greenhouse gas emissions could come from microbial community changes, but another 30% in inefficiencies can be directly attributed to animal stress. So in this case, with slick, you have a trait in which you can ameliorate heat stress and thus increase those efficiencies in these animals. And the main key here is that we're talking about animals that we're importing into countries normally where they weren't developed, not editing within the local native animals themselves for increasing production. So heat stress has very obvious signs of phenotypes for the animals. And a lot of that is in they stop eating. And when they stop eating, they stop production. It also has unseen effects on fertility. So if we want to increase an animal's heat load index, and we can look at different things that we can do, of course, is we can try and make their environment better through agricultural engineering interventions. And a lot of high-end dairies have done that in very hot zones. But we can also change the animal's genotype. And so breeders have been doing that here in the tropics through crossbreeding and making composite animals, but with gene editing, we can do that directly on the animals that were developed in the temperate zones and adapt them straight away. So one way we could do that to remove that heat stress load right up front is just reduce the animal's normal core body temperature. And here I'm showing a graph of the slick phenotype animals versus wild type. And you can see that the average daily body temperature all day long, as the temperature changes, is typically a ½ degree to a full degree Celsius lower than the wild type animal in the tropical zone. So this is temperatures taken at a Florida dairy from lactating animals. This is work done by Pete Hansen. So do we have naturally occurring traits that are responsible for this slick? So the most famous one was, of course, what's called slick 1 now, which was found in the centipole animals. I worked on that in 2009 when I was back with USDA. We continued that work looking at a number of different Criollo breeds. It was interesting because as soon as we found the slick 1 mutation, we looked at some of the other Criollo breeds and they didn't have it, but they were obviously slick. So we went through and characterized the prolactin receptor on a number of different Criollo breeds. Some of that work I did with Marcus Vinicius on Caribou to find slick 2. So what did we find? We found a case of convergent adaptation where we have found now 6 different naturally occurring mutations, all of which truncate prolactin receptor. So prolactin receptor, at least in cattle, seems to be the primary signaling mechanism whereby the animal is told it's going to be hot and that it needs to adapt or prepare to have a heat overload. In Holstein animals that are actively lactating, they can actually start to feel that heat stress at a thermal heat index of as low as 72. So that just means it's very easy for them to enter into heat stress. So we wanted to, when I was still with USDA, we were looking what is the phenotypic advantage as far as milk production. on an animal that carries one copy of SLIC. So SLIC is dominant, so you get the effect just by having one copy and one wild type copy, versus those Holstein animals that don't have that copy. So in Puerto Rico, there's a naturally back-crossed herd that was derived from Criollo cattle, and they imported in Holstein semen and used it over the past 70 years to create basically conventionally bred SLIC animals. There's also Jerseys as well. And so in each one of these bars represents 122 different animals. On the wild type, you can clearly see that there is more of a lactation depression caused by heat stress. And so that difference in milk production is about 10%. What we didn't really expect, and we probably should have, since we knew that the heat stress affects on fertility, but there's a reduction in days to conception on the animals that carry one copy of SLIC. So they breed back faster by 30 days on average. I should say that there's a number of new studies that will be coming out based upon the use of semen from this bull that was developed by back crossing to introduce the Centipole allele into Holstein at the University of Florida. And those are studies done by Pete Hansen and Ann Denicol also at California Davis. So I think they've already done the first study on the calves, whether or not the calves go through heat stress in Florida versus California, SLIC versus non-SLIC. And that paper is published, I believe. And there'll be more coming out as these animals go through lactation. So we introduced SLIC in 2018 into an Angus and actually cloned that animal here in Brazil. Of course, an N1 is not very big, but she was clearly resistant to the heat. And later on, we got funding not only from FFAR, but also from the Bill and Melinda Gates Foundation to create SLIC beef animals and also SLIC dairy animals. And we had our own internal program to make SLIC Angus. Initially, we tried doing it through microinjection of embryos. We found that's not a method that's very standardized. It worked very well. We have now more than 19 different animals that are SLIC. Most of those are at University of Florida and under a grant study. There's some Jerseys there and some Angus. We also had a couple of Angus born before that project started. But we thought the best way to standardize introducing SLIC was actually going to be through cloning. So we skipped the generation of having to breed out mosaic animals. We're putting some sort of a disclaimer on any products we have that not every sperm or egg carries the SLIC allele, and going straight to homozygosity with these animals in a very standardized way. So we're actually going through an ISO process to standardize this. But in here on this diagram, I'm showing that the method that we've chosen is we do high value IVF matings. In this case, I'm showing Holstein sire and dam. From those IVF embryos, we create cell lines, embryonic cell lines. So we're actually going to clone from the embryo from an animal that's never been born before. But we can look at what the genomic value of that animal is before we actually do the editing and the cloning. So we take the embryonic stem cell lines, each individual one that's produced from the embryo, and we do genotyping through Zoetis. We get our genomic merits on that. And then we can pick the best cell line based upon genomics for the editing. We take it through our editing process, which is standardized at Acceligen, to introduce the SLIC trait, and then we clone those embryonic stem cells. And here I'm showing a calf that was born in March, and this animal is top 1% for net merit and plus 2,300 for milk. So some of the pros for doing it this way, of course, is the longevity and culture. We can do multiple edits or multiple edits in a single round. And then, of course, we're using next generation genetics, which is very, very good for the dairy industry. So you can keep in step with the progress that they're making. Here's another calf that was born right here. This animal is not quite as high in net merit, but much more increased in functional traits relative to fertility and health as far as its indexes go. Both of these animals now are over 5 months old and doing quite well. So just take a look at this animal. This is a clone of this one, same one. What's the difference? This animal has 2 additional edits where we put in some alleles that are from our population genetics analysis are exclusive almostly in West Africa. We find them in the breeds that are resilient to trypanosomiasis. And so we've introduced edits in 2 genes within the OXPHOS pathway. We know that the parasite involved in sleeping sickness in humans and Nagana in cattle scavenges heme from its host. And so these genes are within that pathway and they're coding mutations that are not found typically in any species. So they're very unique. So this is the first multiplex animal, we believe, that's been generated and cloned from embryonic stem cells. So that animal was born in July. It's doing well. And we have a couple additional animals as well, which I'm not showing yet. So to change over to beef, how do we address that with Slick? We've focused on Angus because it makes up 87% of the seed stock market in the United States. Of course, it's well known for its high quality beef, its marbling, its tenderness, and its branded beef programs are very well known. The problem is neither of these breeds carry alleles really for heat tolerance. So what has been done, at least in the Gulf states in the United States, is they've introduced Brahman and created ultra blacks or Brangus. So that's typically what you're going to find in the southeast United States. And those animals typically don't qualify for branded beef programs because it's very hard to get those animals up to prime in a timely manner. So Slick Angus we see as a entry into the market, would replace these less efficient and less desirable beef quality crossbreds. And the Slick Angus would actually be eligible for those beef programs. But the biggest market of all is probably here in the tropics, Brazil in particular, because it's a white cow herd numbers about 60 million. We saw a great talk today about how fixed time AI is changing the scenario to create more high quality beef in Brazil, using a lot of semen. So Angus semen sales keeps ramping up and increasing every single year here in Brazil. I think of the 28 million straws, 14 million were imported from the United States, and the majority of that was Angus semen. So this F1 is the Brazilian Angus, and it qualifies for Angus certification. And so really this is the biggest market as far as being able to use a Slick Angus. We think that it would fit very well within this scenario. Currently, from the 1 billion kilograms of beef that's being produced by fixed time AI, those animals reach slaughter anywhere from 3 to 6 months earlier. So what does that mean in savings as far as carbon footprint? It's quite incredible. It's 180 days less to market on average. Land use is reduced, similar to the size of a country of El Salvador. You have about 20,000 Olympic swimming pools saved of water. And then you have this huge reduction in greenhouse gas emission from just using these F1 Angus. So now you can imagine if you put on top of that the ability to put bulls in the field to double the number of F1 Angus that are out there. These bulls should be able to survive in the Brazilian environment. We know that because there's more than 80,000 centipole right now in Brazil, and those bulls are walking around and servicing cows quite nicely in this environment. We also, it's an uncharacterized outcome yet, but the F1 having Slick on one side and that Angus genetics being better adapted may also do a lot better in the feedlot or even in the field relative to the conventional Angus crossed with the Nilor. So that data still has to be generated. So we have a commercial path and we have animals approved for that. So in the United States, we got a low risk determination, which allows us to commercialize both the semen and eggs. And if we wanted to eat these animals, we could without any labeling or need for regulation of that food. This guy, Stay Cool, is actually at Rocky Mountain Siders being collected with certified semen so that we can make exportable embryos to bring here to Brazil. And this female, who's unrelated, is at Transova, and she's in the OPU program. So these are the animals from the limited enforcement discretion, and we've actually applied for the entire trait for both dairy and beef because now we have multiple animals and a lot more sequencing data to back up the fact that we can standardize this process. And of course, in December of last year, it was decided these two animals were non-GMO by CTN Bio. So I'll stop there. Thanks for your attention.